Hello and welcome to this podcast. In this podcast, we're going to talk about protein folding. The importance of it, how it happens, what happens when it doesn't fold properly. We're going to talk about how we have this linear piece of protein and how this will eventually fold up into a mature protein. As we begin this discussion, I want to talk about 4 important bonds that occur to allow us to go from this linear structure to this fully folded protein. The first are disulfide bonds. Disulfide bonds occur between 2 sulfur groups. So if you remember from the previous podcasts, there were only 2 amino acids that had sulfurs in them, cysteines and methionines. However, only cysteines will form these disulfide bonds. The next bond that I want you to keep in mind are hydrogen bonds. Hydrogen bonds, as we form these structures here, are going to be very important in both these intermediate steps and the final structure here. And the amino acids that are going to be important in hydrogen bonds are going to be the polar amino acids. So any of the polar amino acids could participate in hydrogen bonding. Electrostatic attractions. The amino acids that are going to play a role in this are going to be the positive and negative amino acids. So for the positively charged ones, we're talking about lysine, arginine, and histidine. And the negative ones, we're talking about aspartic acid and glutamic acid. The fourth type of interaction that's going to be important in going from this stretch of amino acids into this fully folded protein are van der Waals. And remember, these van der Waals occur because of these very temporary positive and negative charges that exist because of the way the electrons spin around the outer shell of the atom. And lastly are hydrophobic interactions. The tendency to get those hydrophobic residues, rather the nonpolar ones, into the middle of the protein and allow the water in the aqueous solution to interact with these polar residues. Now, I've alluded to this before, but I want to talk about the levels of protein folding. The first one is called primary. What the primary structure is just that linear sequence of amino acids. So from the N-terminus to the C-terminus, you have 1 amino acid, 2 amino acids, 3 amino acids, and all the way down until you get to the N. As we've discussed before, each of these amino acids are held together by peptide bonds. So we would say that the primary structure is characterized by peptide bonds. It is also a non-functional structure, meaning that for that protein to have function, it has to go beyond this linear array of the amino acids. It needs to fold more. But ultimately, the sequence of the amino acids in this primary structure ultimately dictate how that protein will ultimately fold in its final conformation. The next level is secondary. There are 2 main kinds of secondary structures. I'm going to write them here: alpha helix and the beta sheet. Both of these secondary structures take advantage of hydrogen bonding between amino acids that are near them. Not necessarily right next to them, but in the vicinity of each other. We could draw an alpha helix like this. It forms just a simple helical structure. And again, it is characterized by hydrogen bonds between amino acids that are relatively near. So this alpha helix is often found in membranes, or membrane proteins, I should say, that function in transport. Beta sheets can be drawn sort of like this, where they have these distinct edges to them, like so. And they're connected with each other by these linker pieces of amino acids, like so. Like the alpha helix, they're characterized by hydrogen bonds. Again, local interactions to form these kind of steps here. But they will also form hydrogen bonds between different beta sheets. These structures that are formed, sometimes when they're stacked like this, we call them beta pleated sheets. And what they do is they add a lot of rigidity for the entire protein, or at least for that part of the protein. They also often form the core of the protein. They're very stable units. If you think about silk fibers get their really unique tensile strength because of beta pleated sheets like this. I'm going to erase this now so we can talk about the next level of protein structure. The next level of protein structure is the tertiary level. Remember, you had your primary level here, and this primary structure then formed alpha helices, maybe some beta sheets, and most of these interactions occur due to the attractions or the bonding of amino acids in the relative area. As we move to the tertiary structure, these secondary structures will now fold upon each other, and there's often several of them. Maybe more alpha helices, maybe we're going to get a beta sheet here that'll form on itself. Something on these lines here. And these are held together by a variety of bonds, including hydrogen bonds. In fact, almost everything here. Van der Waals, electrostatic, and perhaps the most important, it's hard to say one's more important than the other, but this is really critical for the folding of this tertiary level of the protein. And that are these hydrophobic interactions, where the necessity to place these nonpolar amino acids in the middle of this protein, due to the fact that the aqueous solution around here is interacting with these polar residues, pushing those nonpolar residues into the middle of the protein. Often, these are stabilized by cofactors. Cofactors are often metals like zinc, iron, copper, that will bind here. And they will stabilize these interactions to produce the most stable structure of that folded protein. Other factors to think about, so these are cofactors that can bind, but the ultimate folding or unfolding of this protein can also be influenced by pH, temperature, and salt. At the right concentrations or values here, they help stabilize this structure. Changing these can help destabilize this structure. Typically, the tertiary level is the functional level. The last level here that I want to talk about is the quaternary structure. And what this is when you have one tertiary structure like this one here, or we might call it a subunit, would be a better term, interacts with another subunit over here. They can be the exact same protein as over here. It could be a different protein. So here I'm going to put subunit 2, and over here is subunit 1, because they're different looking at least, right? When we have two interacting with each other, we say that is the quaternary structure. When it's two, more specifically, we would call that a dimer. If there are three, we call it a trimer, 4, a tetramer, and so forth. If they're different, like we see here, they would be called heterodimer, heterotrimer, heterotetramer. If they're the same, we would call them homodimers, or homotrimers, or homotetramers. These quaternary structures are also stabilized by these same kinds of interactions, and they are also considered a functional form of the protein. Some proteins only need a single subunit to function, whereas others require 2 or more subunits to function. The next thing I want to talk about are protein domains. So to talk about this, I'm going to draw different structures of a protein, and I'm not going to draw the specific secondary structures this time. I'm just going to draw them as kind of these globs. Now, I drew them in 3 distinct colors here, even though this is a single protein that I drew. One protein. But this protein has 3 domains. So it has 3 domains. Let me write that here. Each domain behaves independently. They can fold independently of the other to. They can have different catalytic abilities or purposes. For instance, maybe this purple domain has a DNA binding function. Maybe this domain has a substrate binding, substrate other than the DNA. Maybe it's a substrate that binds here and it directs it to the to a different location in the cell. And maybe this middle one here, once it's bound to the cell, the DNA or somewhere else in the cell, it has a third catalytic function. I want to remind you that since this is a single protein, it's a single gene that codes for it. One gene with one start, one stop, makes one big protein. They each fold independently as shown here. They each have independent functions, though they're related. This binds the DNA, this binds a substrate, say, to take it to the nucleus so it can bind the DNA, and maybe this has some catalytic function in like transcription or replication. Okay, that brings us to the end of this podcast, and so let's talk about a quick summary here. In the previous podcast, we had talked about amino acids and categories of amino acids. But in this podcast, what we focused on was protein folding. In discussing protein folding, we talked about the 5 bonds that were important in going from this linear protein, the primary sequence, into this fully folded tertiary sequence or quaternary sequence. We then looked at the levels of protein folding. We talked about the primary level, the secondary level, the tertiary level, and the quaternary level. We talked about, and you should be familiar with, the bonds that help characterize each of these levels, their functionality, which ones are functional in their fully folded form as a primary, secondary, tertiary, or quaternary structure. And we also talked about domains and how a single protein coded by a single gene could have multiple functional domains within that single protein. Okay, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.